Hello everyone. So in this video, let us start discussing the rest of the topics. So first I would like to discuss a very important topic, that is Warburg effect. See, when even though we have ample oxygen, the cancer cells will demonstrate a distinct form of cellular metabolism which is characterized by increased glucose uptake and increased conversion of glucose to lactose, that is fermentation via the glycolytic pathway. This is what we call it as Warburg effect or aerobic glycolysis, because this was invented or discovered by Otto Warburg, for which he has been awarded Nobel Prize. So this is nothing else but what we call as the glucose hunger of the tumors. This is what we call it as glucose hunger of the tumors. This glucose hunger of the tumor has been clinically utilized to visualize tumor via what we call it as PET scan, that is positron emission tomography, in which the patients will be injected with a dye called fluorodeoxyglucose, which is a non-metabolizable derivative of the glucose, which will be preferentially taken up by the tumor cells, and this actively dividing cells such as the bone marrow, everything will be taken. So most of the tumors are PET positive, and the rapidly growing ones are extremely positive for in PET scan. Yes. So the aerobic glycolysis, it will provide instant metabolic intermediates for the rapidly dividing tumor cells. Whereas the mitochondrial, the usual pathway, that is the glycolysis, Krebs cycle, and finally oxidative phosphorylation. So this pathway, it will take a lot of time. At the same time, aerobic glycolysis or the Warburg effect has known to produce or provide instant intermediates, metabolic intermediates to the rapidly growing or the dividing cells. That is a very important point you have to remember. And remember, so what is the mechanism of this Warburg effect? Just remember two important mechanisms. One is what we call it as PI3K AKT signaling, and next one is called as receptor tyrosine kinase activity. So already the PI3K AKT signaling pathway. This pathway has been known to upregulate the activity of glucose transporters and multiple glycolytic enzymes. As a result, it will increase the glycolysis, and there will be shunting of mitochondrial intermediates to the pathways, which leads to lipid biosynthesis, and at the same time there will be enhanced protein synthesis also. Then coming to receptor tyrosine protein kinase. This is the pathway. See, you can see in this picture, this single picture you can draw. Please use these pictures. Very important. Yes, see the glucose, you have the PI3K AKT signaling pathway. There will be increased protein synthesis, nucleotide amino acid synthesis, lipogenesis, and growth, and there will be shunting. So what will happen? There will be more lactate production. And at the same time via MYC pathway. This is another pathway. I told there are two important pathways. One is what we call as the PI3K AKT pathway, and there is the receptor tyrosine kinase pathway. I will tell the receptor tyrosine kinase. See this receptor tyrosine kinase growth factor. This also will cause increased glutamine utilization. This will also shunt the mitochondrial metabolites to the other pathway. That will lead to enhanced growth and protein synthesis. Fine. Yes. So the MYC is also important. Upregulation of MYC, which will causes what required for the mitochondrial utilization of the glutamine. So these are the three important pathways. So what is the number one pathway? PI3K AKT pathway. Number two is the receptor tyrosine kinase activity, and number three is what we call it as the MYC pathway. Number three is what we call it as the MYC pathway. Yes. So this is the PI3K AKT signaling pathway. And the role of MYC gene as well as the role of receptor tyrosine kinase. So what do you mean by autophagy? Remember children, autophagy is nothing else but it is a state of severe nutrient deficiency in which the cells not only arrest their growth but it will cannibalize. That is the cannibalization of the own organelles, proteins and membranes as carbon sources for energy production. That is called autophagy. Remember, autophagy is one of the most important mechanism which has been demonstrated in tumor cells. Just remember this pathway. And next I would like to tell an important update. Important update in Robbins 10th edition. That is what we call it as ongometabolism. What do you mean by this ongometabolism? See, normally there is an enzyme, we call it as isocitrate dehydrogenase, which takes place in the which plays a very crucial role in the Krebs cycle. So whenever there is a mutation in this isocitrate dehydrogenase enzyme, what will happen? Usually the alpha ketoglutarate, it will produce what other metabolites. Am I right? That is succinate CoA. This succinyl CoA will further continues with the Krebs cycle. Whereas in the mutant isocitrate dehydrogenase, one second. Yeah, the mutant isocitrate dehydrogenase, it will causes the conversion of alpha ketoglutarate to 2-hydroxyglutarate. This 2-hydroxyglutarate has been called as an ongometabolite. This has been called as an ongometabolite, and this 2-hydroxyglutarate is known to cause some epigenetic alteration like methylation and histone modification, which causes altered cancer gene expression and cellular transformation. So ongometabolites, it can be a potential short note question because it is a major update in the Robbins 10th edition. Okay, yes, please go draw this flowchart. So what is the basic mechanism? I told you, you know what is normal happening. Normally what will happen when there is isocitrate dehydrogenase, you know. And the second thing, whenever there is a mutation in this isocitrate dehydrogenase, the alpha ketoglutarate will be converted to 2-hydroxyglutarate, and this will in turn causes what? A methylation and histone deacetylation that is responsible for epigenetic alteration and cellular transformation. And here comes a question. This mutant IDH acts as a producing which produce 2-hydroxyglutarate has been considered as a prototypical ongometabolite. Okay, why? Because ongometabolites, they are called ongometabolites. We have oncogene, oncoprotein, ongometabolites because these metabolites are going to enhance cancer gene expression, and these has been included in these oncometabolites has been, or oncogenetic isocitrate dehydrogenase mutation has been implicated in the pathogenesis. So some important carcinomas. Number 1 is what we call it as glioma. Number 2 is what we call it as acute myeloid leukemia, sarcoma, and another one is last one is cholangiocarcinoma. One question must, okay, MCQ, you will get this, all of the following oncometabolites plays a major role in the pathogenesis, except, so you have to remember cholangiocarcinoma, gliomas, acute myeloid leukemias, and sarcoma. Okay, I hope you are clear with this. Fine. Yes. Okay, so that's about Warburg effect. 1 second. Okay, that's about Warburg effect. Once this is done, next important hallmark is evasion of the programmed cell death or apoptosis. Here and out, okay, you won't get much questions like short notes or anything. We are going to just focus on some of the important points for MCQ exams. Okay, fine. So, what do you mean by anoikis? See, anoikis is nothing else but what we call it as, see, there is an important terminology here, anoikis, that is loss of adhesion to the basement membrane. Okay, we call it as anoikis. This anoikis, that is loss of adhesion to the basement membrane, can trigger apoptosis. Okay, yes, so apoptosis is the barrier. In order for the cancer to develop, it should what? Escape from apoptosis. You know the normal pathway of apoptosis, no? Intrinsic and extrinsic pathway. Extrinsic via CD95, intrinsic via, you know, BCL2, BCL, XL, and MCL1. Okay, all this will be over. That is, all these are nothing else but what we call it as what? Anti-apoptotic proteins. Am I right? This anti-apoptotic proteins. So whenever there is an increased anti-apoptotic proteins, what will happen? That is, there won't be any there won't be any apoptosis. Am I right? BCL2 So what will happen whenever there is an upregulation of this anti-apoptotic factors? So anti-apoptotic, that means which inhibits apoptosis. So there will be decreased apoptosis. Fine. Yes. Okay, so next is another important thing, intrinsic pathway. I have told you BCL2, BCL, XL, and the MCL1, they are called as anti-apoptotic proteins. The cancer cells will upregulate the anti-apoptotic proteins so that apoptosis won't happen, and thus they can escape from apoptosis. You know, caspase 3, 6, 7 are called as executional caspases. There is no nothing more you have to remember here. Okay, and just remember BCL2, MCL1, and BCL-XL. Fine. Yes, then CD95, you know, flip proteins, it will prevent the activation. Viruses have flip protein, it will prevent the activation of caspase 8. Then what are the properties because of which the cancer cell has some stem cell like properties? Okay, see, the number one important thing is that the cancer cells have the highest what enzymes, that is what we call as a telomerases. What is the enzymes? Telomerases, whereas the somatic cells do not express this enzyme called as telomerases. Fine. Yes. Then okay, there is something called as the most normal human cells know the capacity to divide 60 to 70 times, and this is what we call it as Hayflick limit. What is this called as? Hayflick limit. This is what we call it as Hayflick limit. Okay, yes, Hayflick limit. And but cancer cells won't follow this Hayflick limit. That means they will divide more than 70 times. Fine. Yes. So the cancer cells have very high telomerase enzyme. So that is also responsible for the continuous division. And because of that, what high amount of telomerase enzyme present, they behave like stem like cells. Okay, stem cells. Fine. Yes. Then self renewal. Okay, you know all these things. Fine. Cancer stem cells. Then next most important point. See. Self-renewing capacity, the best example here they have explained is PML rara. That is, PML rara has been implicated in the pathogenesis of translocation 1517 acute promyelocytic leukemia, so which will produce some mutant stem cell like properties, okay, and that is responsible for self-renewing population. Fine. And next is what we call it as angiogenesis. What do you mean by angiogenesis? Unless and until there is increased vascularity, there will be what the tumor cells cannot grow. So the formation of new blood vessels is what we call it as angiogenesis. So we have some lot of angiogenic and anti-angiogenic factors. Okay, there is always a balance between this. Okay, yes. Okay, so whenever there is a relative lack of oxygen due to hypoxia, which will stabilize the hypoxia-inducible factor 1 alpha, which is an oxygen-sensitive transcription factor, which will activate the transcription of the pro-angiogenic cytokines, VEGF, that is vascular endothelial growth factor, okay, and fibroblastic growth factor. It will express the ligands of Notch signaling pathway, which will regulate the branching and density of the new vessels. Remember this question has been asked, what is the signaling pathway activated by the VEGF? Answer is Notch signaling. Okay, yes. And it will also inhibit some molecules like, that is, thrombospondin 1, and thrombospondin is an anti-angiogenic. So when thrombospondin, which is an anti-angiogenic molecule, is inhibited, that will also again promote VEGF like molecules. Fine, yes. So we have a drug called bevacizumab. Bevacizumab is a monoclonal antibody that neutralizes, that inhibit the VEGF. So when VEGF is inhibited, there won't be any angiogenesis, and it has been used in the treatment of multiple cancers. One of the most important indication of anti-VEGF drugs is in case of proliferative diabetic retinopathy. Fine, yes. Okay, then coming into invasion and metastasis. So invasion and metastasis is a standard short, long question which has been asked in your exam. So first we have to write what do you mean by invasion and metastasis? Fine. What is the two phases which is involved in the metastatic cascade? Number one is we have to breach. We have to break the extracellular matrix. Number two, we have to get into the blood, and finally we have to reach some other places. Okay, that is invasion of the extracellular matrix and vascular dissemination. Then homing of the tumor cells and colonization. So invasion of the extracellular matrix, we have something called as, in extracellular matrix we have two barriers to overcome. Number one is the basement membrane, and number two is the interstitial connective tissue. Okay, yes. So what are the important steps which are involved? First is there is loosening up of the tumor cell interaction, then degradation of extracellular matrix, attachment to the ESCM components, and finally migration and invasion of the tumor cells. Fine, yes. So this is the step. See, first angiogenesis, then adhesion to the basement membrane, passage through extracellular matrix, intravasation, that means getting into the blood vessel, interaction with the host lymphoid cell, tumor cell embolus, adhesion to the basement membrane, extravasation, metastatic deposits, angiogenesis, and finally growth. Out of that, E-cadherins. E-cadherins is one of the most important things. See, E-cadherin helps in attachment of the cells in the basement membrane. Whenever there is a loss of E-cadherin, what will happen? There will be loose adhesion between the cells, so the tumor cells can very easily penetrate and breach the basement membrane. They can enter into the systemic circulation. That is why loss of E-cadherin is implicated in the adenocarcinoma of colon, stomach, and breast. In all this, E-cadherin function is lost. Okay, yes. Then the second step is degradation of the basement membrane and interstitial connective tissue. Okay, so you can see in this picture also. Okay, there will be loosening of the intracellular junction, degradation of ECM with the help of type 4 collagenase, and finally migration and invasion. Okay, yes, fine. So matrix metalloproteinase 9, which is a type which is gelatinase which cleaves type 4 collagen. Okay, it will stimulate the release of VEGF. And the third step is nothing else but attachment of the tumor cells to the extracellular matrix protein. What are the proteins they will get attached? They are mainly attached to the integrins and the fibronectins. Okay, integrins and the fibronectins. So after getting attached, the locomotion is the final step of invasion. What do you mean by locomotion? They have to go to some other places. No, yes, that is mainly done with the help of a receptor called as tyrosine kinase MET. Tyrosine kinase MET. Just remember the key points, because you are these much things. First, when they ask us a long question, first write what are the two phases, then put the heading, please mention these 4 points, then put this flow chart first, then try to explain each of the headings. Fine, that's what you are going to do. Then I told loss of adhesion to the basement membrane, we call it as anoikis will happen. And remember I told you before it undergo metastasis, there will be some interaction with the lymphoid cell. And what is the adhesion molecule or the interaction molecule? It is CD44. This has been asked as an MCQ question. Okay, CD44. I'm just highlighting the important points. Okay, yes. And then finally the site. So the tumor cells, for example, the patient is having breast carcinoma. It should where it undergo to which place, okay, to which organ it should undergo metastasis. It depends upon the affinity of the particular tumor cells. For example, prostatic carcinoma has a affinity towards bone, bronchogenic carcinoma towards the adrenals and brain, neuroblastoma to the liver and bones. So all this, it depends upon the tumor cells only. Okay, yes. And the next most important point that I have to mention here is what are the genes? Okay, what is the genes which helps in? So the metastatic oncogenes are nothing else but what we call as the snail gene and the twist gene. We have two important genes that is responsible to promote epithelial to mesenchymal transition. Okay, fine. And this has been expressed in lot of carcinomas like breast carcinomas, gastric and colonic carcinomas. Fine, yes. Then I have told you what is desmoplasia in the first class itself. Then next is evasion of the host defense. Out of that, here that's not that much important points you are having. Out of that, we have something called as MAG. Okay, that is melanoma antigen gene. Okay, yes. So this, what is the importance of this melanoma antigen genes? Melanoma antigen genes are nothing else but it's a family of what antigens? Okay, it's a family of antigens which are expressed by a variety of types of tumor, most like lung, liver tumor, and stomach and esophageal carcinoma. And what is the most important purpose of these genes? Overexpression of this predisposes to cancer. Whenever there is an overexpression of this major family of antigen, there will be increased risk of getting the above mentioned cancer. Then what is tumor antigens which are produced? We have a lot of tumor antigens, Epstein-Barr virus, human papilloma virus antigen. Yes. Then all this also causes increased risk of cancer. Then oncofetal antigens. Oncofetal antigens are proteins that are expressed at high levels on the cancer cells and in normally developed fetal tissues. We have oncofetal antigen that we are using, that is human chorionic gonadotropin, alpha fetoprotein. These are used as tumor markers. Okay, tumor markers. See, alpha fetoprotein and even carcinoembryonic antigen. That is in case of colon and pancreatic cancer. Okay. Then next is what we call it as MUC1. What is MUC1? It is an integral membrane protein that is normally expressed only on the apical surface, a site that is, there are some certain regions in our body which are sequestered from the immune system. One is our testis, that is seminiferous tubules. Number two is the lens proteins. Am I right? Yes, in the eye. And the third one is MUC1, which is a membrane protein which is expressed only on the apical surface of the breast ductal epithelium. Okay, and this has been expressed in both ovarian and breast carcinoma. It can be used as a tumor marker. So this has been asked as an MCQ. MUC1 is expressed by what cancer? Breast and ovarian cancer. CA125, I have already told you it is ovarian cancer. CA99 and also I have already mentioned in the tumor markers. You can see that lecture. Fine. Yes. Next is what we call it as CD30. CD30 is a member of TNF receptor family. This is mainly expressed by most of the T cell lymphomas and Hodgkin's lymphomas. Then NK cells and cytotoxic T lymphocytes are the two important lymphocytes which have very potent action against destroying the tumor cells. Okay. Then immune surveillance and escape. Here most important concept I am going to discuss is CTLA4 or PD1. See, there are some immunoregulatory pathways. Okay, yes, that means the tumor cells. Okay, I will tell you. I have already told about this in the chapter of autoimmunity. Okay, you can watch that lecture also. See, actually, what is happening? The antigen presenting cells. Okay, through a lot of there are some co-stimulatory molecules like CD28 and all. So the tumor cells, through a vast variety of mechanism, it will down-regulate the expression of this co-stimulatory molecules on the antigen presenting cells. So as a result, what will happen? The antigen presenting cells will fail to engage the stimulatory receptor and it will instead activate the inhibitory receptor on the effector T cell. See, remember, normally CD28 is a stimulatory molecule. That means CD28 will promote immune response against a foreign body or a foreign antigen. But the tumor cells will down-regulate the expression of CD28 and it will up-regulate the expression of an inhibitory receptor called CTLA4. CTLA4 is an inhibitory. That means it will inhibit the immune response. That means the cancer cells are using our own immune system to protect. Am I right? It is going to activate our inhibitory receptor called CTLA4 and it is going to get protection. Am I right? Yes. And the tumor cells will also express what upregulate the expression of PDL1 and PDL2. Okay, and this yeah, and what is this PDL1 and PDL2? This is also finally going to activate PD1 receptor, which is again going to inhibit the T cell activation. So what is tumor cells actually doing? They will inhibit CD28. So CD28 will normally activate the immune response. It is inhibited, no immune response. It will activate what? The inhibitor receptor called CTLA4, PDL1, PDL2, which in turn activate PD1. Finally, all this is going to inhibit our immune response. So recently, this is the important point. What is the clinical application? When we develop, currently scientists are trying to develop antibody which is going to inhibit the CTLA4. When we are inhibiting the inhibition, minus into minus become plus. That means we can activate the immune response, so that the immune, this is how we are using our immune system. Recently we have a lot of recent advances and all we are seeing in newspapers. We have a lot of immune modulatory techniques and procedures and drugs we are developing, which we are using our own immune system to fight the cancer cells. Okay, just remember that point. Then genomic instability. As far as genomic instability is concerned, we have a lot of conditions like HNPCC, that is hereditary non-polyposis colorectal cancer, in which there is a DNA mismatch repair. Okay, and one of the most hallmark of such a mismatch repair is called as microsatellite instability. Okay, so microsatellite, mini satellites, microsatellites, you all read in your biochemistry, genomics. Okay, so microsatellite instability is one of the most important hallmark of patients with mismatch repair. And at that time I have told you already, MSH2, MLH1, all these are approximately accounting for most of the cases of HNPCC. Don't confuse HNPCC with the FAP. FAP is familial adenomatous polyposis coli, that is caused because of the APC gene mutation located in chromosome number 5. This is HNPCC, that is MSH2, MLH1. It is a microsatellite instability, and this is a DNA mismatch repair. Xeroderma pigmentosum is nucleotide excision repair. Okay, that is xeroderma pigmentosum, ultraviolet B rays. Then next one is what we call it as, we have other conditions like the defect in homologous recombination, which is going to cause ataxia, telangiectasia, Fanconi's anemia, Bloom syndrome, all those. Okay, yes. Then cancer enabling inflammation, anoikis I have already told you. Then there are some chromosomal translocation. Out of that, overexpression of a proto-oncogene, which is caused as a result of translocation, is exemplified in Burkitt's lymphoma. We have two genes. One is chromosome number 8, which have the MYC gene, and chromosome number 14, which has the immunoglobulin gene. Whenever there is an 8 to 14 translocation and is resulting in amplification, which will be causes what we call it as Burkitt's lymphoma. See, these are the various translocation. Okay, just no need to mug up this as of now, because each of this condition you will see in the in the particular chapter. At that time you can see translocation in CML we have 922, in Burkitt's lymphoma 814, in Ewing sarcoma we have 1122, 721 and 1721 are seen in case of prostatic adenocarcinoma. This has been asked as an MCQ question. Okay, in prostatic adenocarcinoma, Ewing sarcoma, Burkitt's lymphoma, all these are potential MCQ. Mantle cell, all these you can read in the subsequent chapter. Fine. Then we have PML RARA, that is promyelocytic leukemia, which is a variety of acute myeloid leukemia. There is a 15-17 translocation. In 15 we have the PML gene, and RARA. RARA is retinoic acid receptor alpha. Okay, that is located in chromosome number 17. So 15-17 translocation leads in PML RARA. Okay, so retinoic acid receptors drugs can be used in the treatment of PML RARA type of acute myeloid leukemia. Okay. Then we have CMYC and NMYC. I have told you NMYC in case of neuroblastoma, YEP2 amplification in case of breast carcinoma. Okay, we have, okay, what do you mean by chromothripsis? Chromothripsis means what? That means chromosomal shattering. Okay, chromothripsis means what? Chromosomal shattering. Just remember this point. There is no need that you have to understand. Chromothripsis is called as chromosomal shattering. It is in what? Present in osteosarcoma and glioma. Osteosarcoma and glioma has known to exhibit chromothripsis. That means chromosomal shattering. Just MCQ purpose. That's all. Then there are some epigenetic changes which happens that I am going to see. I told you already what is epigenetics. In that, in in SNF5 gene, okay, causes, whenever there is a mutation, it causes malignant rhabdoid tumor. PBRM1, nucleosome positioning chromatin remodeling, that is its function. Whenever there is a problem in that, it causes renal cell carcinoma. And ARID1A, it causes ovarian clear cell carcinoma, and CREB-P, which causes DLBCL, diffuse large B cell lymphoma. Only please go through the highlighted points. They may be asked as an MCQs in Central Institute exam. Then, next is I want to see about onco-MRRs. What are OncoMIRs? What is OncoMIRs? See, they are nothing else but the non-coding RNAs. Okay, we have the non-coding RNAs, right? Yes, non-coding. They are the microRNAs. MIR stands for microRNAs. See, MIR 200 has been shown to promote epithelial to mesenchymal transcription, and MIR 155 has been is overexpressed in many B-cell lymphomas like B-cell lymphoma. So MIR 200 epithelial mesenchymal transcription. Name other two genes that I have mentioned so far which promotes epithelial to mesenchymal transcription: snail and twist. The third one is MIR 200, MIR 155 in various other lymphomas. And remember, MIR 15 and MIR 16 in case of CLL, chronic lymphocytic leukemia. Very, very important. These are the things only mentioned in Robbins. No textbooks can give such recent advanced so high points. Fine. Yes. And remember, Dicer mutation, it has been included in many of the testicular tumors. Okay, Dicer has been seen in case of testicular tumors. Fine. Then okay, this is how a molecular model. See, the cancers won't happen as the result of a single mutation. See, the best example which I can demonstrate to explain that is, see, the case of colonic carcinoma. There will be first mutation will be because of at the APC chromosome 5q21. Following that, there will be second hit at beta catenin methylation, then KRAS, proto-oncogene, finally TP53 SPAT. This is only finally, then only we will getting the colorectal carcinoma. See, we have 1, 2, 3, 4. Okay, yes. So this is the molecular model. This is the same thing again we will discuss in colonic cancer. Okay, yes. So I hope we have discussed almost everything in tumor pathology. So final part also I have covered. Okay, in the next video you can see the final part where I will be discussing about the carcinogenesis. Then next one is what we call it as the the molecular, that is the microbial carcinogenesis, and finally the tumor markers, paraneoplastic syndrome, everything I have mentioned there. So thank you. Hope you would have understood. So this is Neoplasia, is a very vast topic. Please take your time, revise things, revise the points again and again. And as far as your prof exam is concerned, you can focus on those important areas, and rest of things just for your entrance exam points of view or for solving MCQ, you have to read in depth. Okay, just go through the highlighted points that I have. There is no need that you have to sit and read each and every line. The highlighted points you can focus, you can by habit and revise it again and again. Thank you.